Hello friends, this video on Biotechnology Principles Part 15 is brought to you by examfear.com. No more fear from exam. So with this we have discussed about the second tool for genetic engineering, and that was the vector. So now we are going to talk about the third important tool of genetic engineering, and that is the competent host. Now, if we do not have a host, if we do not have an appropriate host, then there is no point of creating the recombinant DNA, because end of the day this recombinant DNA needs to be introduced into the host cell. So that is why it is very important to talk about the competent host. Now here you can see this is the host. So this one is the host and this one is the recombinant DNA. So our aim is to introduce the recombinant DNA inside the host cell. Now DNA cannot pass through the cell membrane of host cell directly. Now the question is, how exactly do we introduce this recombinant DNA into the host cell? Is it possible to introduce it directly? Now whenever you talk about the host cell, what is present as the boundary of the host cell? Boundary is nothing but the cell membrane. Now if you remember the structure of the cell membrane, so you would remember that it has a bilipid structure. So that is, it has two layers of phospholipids present. So if you look at the phospholipid layer structure of the cell membrane, you see that one end of the cell membrane, that is the outer ends of the cell membrane, is hydrophilic in nature. What is hydrophilic? Hydro means water and philic means loving. So that is, they are water loving, whereas the other end is hydrophobic, that is, they are water haters. They do not like water at all, and therefore they do not like water loving substances as well. So this is how the structure of the cell membrane is, and due to this structure of the cell membrane, the recombinant DNA cannot directly pass through the cell membrane. That is because if you talk about DNA, so DNA by its nature is hydrophilic. Now since DNA is hydrophilic, it is very difficult for DNA to cross this membrane, because in this membrane outer layer is hydrophilic but the inner layer is hydrophobic. So it cannot directly pass through the cell membrane. So now the question is, if it cannot pass through the cell membrane, then how do we introduce the DNA inside the host cell? Because it needs to enter the host cell, but it cannot enter on its own. So how do we make it enter the host cell? So you can understand this concept with a very simple example. Let us suppose that there is a big party which is being thrown by this person. So this person is the host of the party, because he is the one who is hosting the party. He has organized the party. Now you are one of the guests and you want to attend the party. But for anybody who wants to attend the party should have a pass for their entry. Now unfortunately, let us suppose you do not have the pass. So let us suppose there is a guest who wants to enter inside, but the guest doesn't have a pass. So if the guest doesn't have a pass, what will happen? Do you think the host will welcome the guest? Now, two scenarios are possible. If the guest has a pass, then the host will welcome the guest because the guest has a pass. But if the guest doesn't have a pass, in that case, what will happen? The host will not allow the guest. So in that case, the guest would need a mediator or another person who has a pass, and then through that mediator, he can enter inside the party. Right? So the same concept applies here. In this case, who is the guest? So guest is recombinant DNA. Now recombinant DNA doesn't have a pass because recombinant DNA is hydrophilic in nature, and due to this hydrophilic and hydrophobic structure of the cell membrane, it directly cannot pass through the cell membrane. So it doesn't have a pass. Now when it doesn't have a pass, it needs a mediator who can give him a pass, and then the host will welcome him to the party. So who is going to be that mediator? So that is what we will see here, that here in this case also, since it cannot enter directly, but it has to enter, it has to attend the party. So there are some other techniques by which it will be able to enter inside the host cell. So here we are going to talk about this mediator. So these mediator here is nothing but some of the techniques which can help in introducing the recombinant DNA into the host cell. So host cell to be made competent to take up the DNA. Now it is not only the techniques which will help the recombinant DNA to enter inside the host cell should also be prepared enough to take up that recombinant DNA. Now let us suppose in the party, example of the party, the host who has organized the party should be aware enough and he should be prepared enough to entertain all the guests who come in, because he should have all the arrangements ready. So similarly, the host cell also has to be competent to take up the recombinant DNA which is going to enter inside it. So now we will discuss about the techniques to insert recombinant DNA into the host cell. Now some of the important techniques which help in this process is gene gun method, electroporation method, micropipette method. So these are some of the very common methods which are extremely useful in inserting recombinant DNA into the host cell. Now we will discuss about each of these methods in detail one by one. So let us start with the gene gun method. The method itself talks about a gun. What is a gun? So inside the gun you have bullets. So when you fire the gun, the bullets are fired. So the bullets are thrown out of the gun at a very high velocity, and wherever they go, they just penetrate through that object and serves the purpose. So that is how a gun works. So in a very similar way, you have something called gene gun. So gene gun is going to be interesting. So let's see what it is. So what happens in gene gun method? So in this method, small particles of tungsten coated with DNA are fired with high velocity into nucleus of host cell. So that is how it is directly injected into the host cell. So you have a gun-like structure where these small particles of tungsten come out with very high velocity. So the velocity is so high that it penetrates inside the host cell. In fact, it gets into the nucleus of the host cell. So without any barrier of the cell membrane, the hydrophobic nature, due to the high velocity at which these particles are fired, it gets into the nucleus of the host cell. Now this process is also termed as biolystics. So biolystics is nothing but the gene gun method. So this is one way by which the recombinant DNA can be introduced into the nucleus of the host cell. The next process could be electroporation method. Now what is electroporation method? Again here you have the term electro. That means it makes use of electricity. So let us see what happens in electroporation method. So let us see what happens in electroporation method. So in this method, it makes use of electric current to introduce recombinant DNA into host cells. Now this is going to be interesting. Let us see how electric current can be utilized to introduce DNA into host cell. So let us look at it experimentally, because only with the experimental setup you will be able to understand it. So let us suppose this is a chamber where you have an electrolyte as well as the electrodes. So let us suppose these are the two electrodes. One is a positive electrode, the other one is a negative electrode, and you have a divalent electrolyte here in this chamber. And let us say that electrolyte could be any divalent electrolyte that can act as a catalyte. So there is a divalent catalyte here inside this chamber. So this divalent catalyte could be anything like calcium ion. So these are the two electrodes as I mentioned, and let us suppose this is the host cell. So inside this host cell we want the recombinant DNA to enter, and let us suppose these are the recombinant DNA which are present inside this chamber. So we want the recombinant DNA to enter inside the host cell. So how do we do that? Now with this setup, we can very easily establish an electric current when we have the electrolyte and we have the positive and negative electrodes. Now we develop an electric current. Now once the electric current is developed, what will happen? A potential difference develops across the cell membrane of the host cell. So if you closely look at the host cell, so this is how it becomes. This was the host cell. So you have a cell membrane like this. So what happens is, as soon as electric current is established, a potential difference develops across the cell membrane of the host cell. So this is the host cell. So now once a potential difference develops across the cell membrane, so what will happen? DNA is negatively charged particle. So we all know that DNA is negatively charged due to the presence of the negative phosphate ions. Now DNA being negatively charged particle, which is currently outside, it will get attracted towards the positive charge, and positive charge is present towards the inner side of the cell membrane. Now as a result of this particle will tend to get inside. So as a result of this difference in potential difference, the host will take up the recombinant DNA. Now, how the DNA enters? It enters through the pores which are present on the cell membrane. Now, on the cell membrane also you have pores like this. So through these pores of the cell membrane, the DNA enters, and it enters under the influence of this potential difference, and this potential difference arose due to the development of electric current. Now in this way, the host takes up the recombinant DNA. Now as soon as the host takes up the recombinant DNA, the host becomes genetically modified. So this is how recombinant DNA is pushed inside the host cell with the help of electric current, and that is why it is called electroporation method. Electro from electric current and poration from pores, because under the influence of electric current, the recombinant DNA enters inside through the pores present on the cell membrane. So this is all about the electroporation method. Now let us look at the third technique, which is the micropipette method. So what happens in micropipette method? So in this method, micro means very small, pipette means the pipette is like an injection kind of a thing. You would have seen it, like for example in your nasal drops or any other drops, you would have seen the dropper, right? How it looks like. That's a pipette. So that's how a pipette is. So a very small pipette is used to directly inject recombinant DNA into nucleus of the host cell. Like this process and the gene gun method are similar processes, just that in gene gun you have a gun which actually throws the particles at a very high velocity, and those particles are coated with DNA. And in this case, you have a pipette which will directly inject the DNA into the nucleus of the host cell. So micropipette is used for this purpose. So here you can see how a micropipette looks like. It is almost like an injection, like how you have an injection. You put it inside, right? It just gets inside your body directly. So that is how the concept of micropipette works. So these are some of the techniques with the help of which recombinant DNA can be introduced inside the host cell. This is also known as micro-injection method because we are directly injecting it. So it is called micro-injection method. Thank you. Please visit www.examfear.com to watch more educational videos with a better experience. Please do not forget to like and subscribe to our YouTube channel for latest updates. Thank you once again.